Welcome to the So That's Why podcast, where we unpack the science behind everyday questions. I'm Jen.
I'm Chris.
And I'm Matt.
So before we start, a quick heads up about today. Today is about the biology, what physically changes inside a cell to turn it cancerous. So we're not going to be looking at causes, risk, or anyone's diagnosis. So if this is a heavy subject for you right now, it's completely okay to sit this one out.
Yeah, and I think it feels important to say that plainly, doesn't it? Because cancer really is a heavy topic. But it's something we get asked about all the time. So today we're aiming to get stuck into the science, really because the related cell biology itself is one of the most extraordinary stories the body has to tell.
Which is good, because I'll be honest, this is one of those topics I've steered away from a little bit. And a lot of people, I think, do the same. It feels like a massive topic, doesn't it?
Yeah. And I think that's really honest, and it's part of why we wanted to do it. So here's the question. Your body will make tens of billions of new cells today alone. And to make each one, it copies 3 billion letters of DNA. And almost every copy comes out flawless. So the real puzzle isn't why cells sometimes go wrong. It's why they so rarely do. But why do cells turn cancerous?
Yeah, I think before getting into it, that scale you mentioned there, Jen, it's so important to keep in mind. So over a lifetime, that's tens of trillions of cell divisions, each one copying what's effectively a 3 billion letter instruction manual.
Which is crazy.
Can't get your head around it at all.
Right. I mean, that would take a supercomputer.
Yeah.
So, because my body is doing this sort of copy job, isn't it? This sort of big copying, and it's mostly getting it right, which is, I mean, I can't copy a phone number without messing up a few of the digits. So how my body does this, I have no idea.
I know, honestly, I'm the same. But you know, it's the right instinct. So errors do happen. But the remarkable part is actually how much proofreading and repair sits on top of it all to catch them before they even matter.
Which, let's be honest, this is really what it all comes down to. So cancer isn't one dramatic event. It's what happens on the very rare occasions where a series of those safeguards fail in the same cell, one after another.
Okay, so let's start with what a normal cell does, because you can't see what's broken until you know the rules. So a healthy cell only divides when it's signaled to. It does its specific job, and here's the one people forget or maybe don't know. It dies on a schedule too.
So my cells are programmed to die. Sounds a little bit like a fault, not a feature, not going to lie.
Actually, the opposite of a fault. And it's called apoptosis, which is a controlled self-shutdown the body uses to constantly clear out cells that are old, that are damaged, or that are no longer needed.
So apoptosis sounds like a pop group, isn't it?
It does.
Apoptosis is a cell then doing the responsible thing and stepping aside when it's supposed to.
Yep. And that's the deal that a cancer cell breaks. So it stops obeying any of those rules, not because anything invades from the outside, but because its own DNA instructions get rewritten.
That's it. And to see how that rewriting happens, I think it helps to know what the instructions are made of and how they get copied, doesn't it?
Yeah, well, go on then, because DNA is one of those words I use confidently, but cannot really ever explain. And don't ask me what it stands for. I just would not know.
So most people are the same. Okay, so DNA, deoxyribonucleic acid.
You're just showing off now.
Yeah, I am.
We can ask you to pronounce that at the end.
Yeah.
It's a long instruction written in a 4-letter alphabet. So you've got A, T, C, and G, and they're also called bases, and they pair up in a fixed way. So A always bonds with T, and C always bonds with G.
So it's two matching halves, and each half tells you what the other one should say?
Yep, and that's what makes the copying possible. So you might have seen the classic DNA double helix structure, and when a cell divides, that double helix basically unzips down the middle, and each strand is used as a template to rebuild its partner one letter at a time.
So the pairing rule is a built-in check then. If one side reads A, the new partner has got to be a T, or something's gone wrong.
Yeah, and that check it's why most of the errors get caught. But across 3 billion letters, some slip through, and they come in 3 different kinds. So you can have them where a letter can be substituted for the wrong one, or an extra letter can be inserted, or one can be deleted.
Okay, and I'm guessing, and the magic scientist that I am, that an insertion or a deletion does more damage maybe than a straight swap.
Well, you get a science point there, Matt, because yes, usually. And it's because DNA code is read in 3-letter blocks, and each block codes for one amino acid. And an amino acid, as you may know, is a building block of a protein. So if you substitute one letter, you can often change just a single block. But if you insert or delete one, every block after that shifts along by one, and it's called a frame shift. And basically from that point, the rest of the instruction reads as nonsense.
A frame shift. Love that phrase. So a substitution is a typo in one word, but an insertion or deletion knocks the whole sentence out of step from that point on.
Right. And any of those, like a substitution, an insertion, or a deletion, is what we mean by a mutation. And that is a permanent change to the DNA sequence in that one cell.
Okay, so if these slip through then often, 3 billion, why aren't we constantly in trouble?
Because the vast majority are harmless, so they'll land in stretches of DNA that don't actually code for anything, or they get repaired within minutes, or the cell triggers apoptosis. So a mutation, it only starts to matter when it hits one particular kind of gene.
Yeah, and that's such an important thing to remember, isn't it, to kind of put it into context. So there are 2 key families that count here, and the first is what we call Proto-oncogenes, and they normally drive healthy growth. So they tell cells to divide when there's a real signal. However, when you mutate one of those proto-oncogenes, it becomes an oncogene, meaning it's locked permanently on. So the cells divide nonstop, even if there's no signal at all.
So the gene whose whole job it is to say grow gets stuck in the forward position, in the on position, still shouting grow when nothing is actually asking it to, I'm guessing.
Yeah, stuck shouting grow. So you mentioned one family. The second family is a tumour suppressor gene. So they do the opposite job. They hold growth back, repairing DNA and calling for apoptosis when a cell is too far gone. But in cancer, those get switched off.
They do, and the most important tumour suppressor is one you've probably heard us talking about elsewhere, and it's P53, which is also nicknamed the guardian of the genome.
I feel like I want to salute it when you say guardian of the genome.
Well, it's important enough. I think it deserves it, because when DNA is damaged, P53 either halts the cell so it can be repaired, or if it's beyond saving, it orders that apoptosis, that cell death. And P53 is faulty in more than half of all cancers.
So losing them P53 doesn't just allow damage, it removes the very thing that would have caught the damage. So it feels like a double blow when 53 is not doing its job.
It really is. And this is why it takes so long. So a cancer cell can carry hundreds of mutations, but most of them are passengers. So just effectively along for the ride doing nothing. It's that handful of driving mutations that matter. And it usually takes 6 or more distinct drivers stacking up in one cell line to kind of do the main damage.
And there's a detail on the tumour suppressors worth having. So you inherit 2 copies of each. So both have to be knocked out before that safeguard is truly gone. It's called the 2-hit rule, and it was first worked out in 1971 by a scientist called Alfred Knudsen.
Not Alfred.
Yeah, Alfred.
Okay.
R.L.
R.L. Yeah. So that's why I guess some cancers may be running families. I'm guessing if you're born already missing one copy, you only really need the second hit rather than the 2, and you maybe are a step closer to the edge.
Yeah, it's true, but I think it's really important to say it doesn't mean anyone's fate is sealed because of that. It just means that the count starts from a different place.
Yeah. And once a few drivers are in, it can start to accelerate because some of the genes that get knocked out are the DNA repair genes themselves.
Okay, I'm not sure I like where this is going. So once the proofreaders are gone, the errors, I'm assuming, then pile up faster.
Yeah, and that's the honest, and I think fair to say the unfortunate picture here, because the whole genome in that situation becomes unstable. it's what's called a mutative phenotype. So more drivers arrive sooner, and a process that crept along for years can appear to speed up.
Which reframes something for me, I guess. So when people say cancer appeared suddenly, it probably wasn't sudden. It had been building up maybe for a long time and had just sort of crossed a line laid on, perhaps.
Yeah, it's true for a lot of cancers. What looks sudden is usually the visible end of a long, quiet process.
Yeah, that's right. And moving around, there's one more layer worth talking about, but I will be upfront that it's newer and it's still kind of developing in cancer research. And this is the concept that cells can also silence genes without changing the DNA letters at all.
Hang on, so you can switch a gene off without editing it. How does that work?
So the main route is a chemical tag called methylation. So the cell adds a methyl group to a gene's control region and it goes quiet. So not one letter is changed. The instruction's still there. The cell just can't read it. And in cancer, that can switch off those same tumour suppressor and repair genes.
So you get the same result as a mutation, an off switch, but with nothing you could spot in the sequence itself.
Broadly, yeah. And it's quite incredible, really, isn't it? Not necessarily in a positive way.
No.
But, I'll stress again, this research is still maturing, but it's clearly part of the picture. But it's fair to say, it's a layer that we understand less completely than the mutation point.
And it's worth also saying cancer isn't one thing. So it's a family of diseases in different tissues that share the same story, which is part of why no single treatment fits all of them.
So even a single tumour, I'm guessing, isn't all identical cells?
Correct. That's usually not the case. As those cells keep dividing, they pick up different mutations along the way. So a tumour is usually a mixture of slightly different versions, and that mixture is exactly why treatments can be so stubborn.
So the therapy then, I guess, clears out most of the cells, and the few that happen to be different seem to survive and grow, and that variety, I guess, is what makes it so hard to beat.
Yeah, it is. And by the time a cell has gathered enough drivers, it's picked up a whole set of abilities a normal cell would never have.
Yeah, and the classic list of those things is sometimes called the hallmarks of cancer. So it grows with no signal, it ignores every stop signal, and it refuses to undergo apoptosis or cell death. So it keeps accumulating.
So it's broken then. It's broken all three of the rules we started with. It grows uninvited, it won't stop, and it won't step aside.
All three, and then it adds more. It becomes effectively immortal. So normal chromosomes have protective caps on them called telomeres that shorten with each cell division until the cell retires. But cancer cells switch a repair enzyme, telomerase, back on to rebuild them.
So the cell reactivates an enzyme that resets its own counter and cancels its retirement. Sounds all a bit futuristic, a bit AI, a bit alien invasion.
Yeah.
If I'm honest.
Yeah, and it's a genuinely strange trick. And there's one more that I'd say is arguably even more sneaky, because your immune system, which is obviously amazing, it's constantly patrolling for abnormal cells. And so a cancer that survives has learned to hide, basically. So it stops showing the surface flags that mark it as faulty, and it puts out checkpoint signals. So we're talking about molecules like PDL1, which is a protein that tells immune cells to stand down.
And there was a study in the Nature Journal in 2025, and that found that it can go further still. So the cancer cell physically passes its own faulty mitochondria, the parts that generate a cell's energy, into the immune cells sent to destroy it, jamming their power supply so that they can't fight back.
So it doesn't just hide from the immune system, it disables then the very cells sent to kill it. Every one of those is another rule it's learned to break then.
That's it, and that's probably the natural place to end that mechanism. So with all these safeguards, the question is, why does the cell not just stop itself? And the important point here, when you go back to those millions and trillions that we talked about, is it almost always does.
Yeah, and that's a bit to focus on, isn't it? Because after all, all what we've talked about is incredibly easy to feel like the odds are stacked against you and terrible.
Yeah, they really aren't though, and that's what we'd want people to take away. Given the sheer number of cell divisions every day, cancer is genuinely rare, so the safeguards catch the overwhelming majority, and it's only the very unlucky pile-up of failures in the same cell over a long time that gets through.
Yeah, and importantly, every one of the steps we've named is now something that research can target, and that's from drugs that block oncogenes to ones that switch the immune system response back on. And so understanding how the cell breaks each rule is what tells us how to step in. And let's be honest, the advances are still incredible if you think where we are today versus even 10 years ago.
Absolutely incredible, yeah.
Yeah, they really are. Okay, so let's bring this home. Our original question, why do cells turn cancerous?
Because a single cell accumulates driver mutations that break the rules it lives by. So the proto-oncogenes that say grow get locked on. The tumour suppressors that say stop, repair or self-destruct get switched off. And the safeguards that will catch all of that fail too. So you stack up enough, usually over years, and the cell stops being cooperative with the body and starts acting for itself.
Yeah, and as we said before, the thing to keep firmly in mind here is that it's rare, precisely because the body is so good at catching it. So what we're describing is the exception that slips through an extraordinary amount of protection.
And weirdly, I find that reassuring rather than frightening, which is perhaps not how I felt at the start of the conversation about cancer.
Yeah, it's definitely helpful to understand the process in this case. So that's why cells turn cancerous. That is it for this week. Make sure you hit that subscribe button and tell us in the comments what questions you'd like us to explore. Thanks for geeking out with us. But for now, that's it from me.
That's it from me.
And that's it from me.
Bye for now.